So welcome to this session, and as we, in keeping in tradition with what we were supposed to do in this session, we are supposed to have a discussion. What I will quickly do is to tell you something, summarize what Vaishnavi was talking about, and give some sort of a perspective as to how to approach the problems that are there in this, in mitochondrial morphology and dynamics, from a physics perspective. So from trying to see how we can look at these systems and see what type of models or how can we understand these behaviors. But before we do so, let's quickly go through what we have what Vaishnavi has been talking about regarding mitochondria and microtubules. So these are these microtubules, and this is the mitochondria that she had shown. Now, these are organelles which are there throughout the cell, and this is the organelle that we are going to focus on. So a brief history about the mitochondrion, and something which has been some questions that were asked during Vaishnavi's talk. It's established. It's no longer, I don't think it's more of not for theory, but it's quite established that mitochondria are descendants of ancient bacteria that entered into a symbiotic relationship with primitive host cells. This theory is called endosymbiotic theory. They actually retain several characteristics of the bacterial ancestors. For instance, they have a double membrane, the proteome similar to that of alpha proteobacteria, and their ability to synthesize ATP via a proton gradient created across the inner membrane. So these are all characteristics that they have carried from the ancestors. Mitochondria also undergo membrane remodeling through cycles of fusion and fission, which of course you have heard in great detail from Vaishnavi. And this balance of fission and fusion controls mitochondrial structure. And depending upon the cell types, you can have situations where there is a single connected structure, okay, mitochondrial structure. So regarding the shape of the mitochondrion, we know that, at least this one is for the mammalian cells, and for this we know that there's rapid changes of shape which happens. This is across 20 minutes, and you can see that it takes these kind of shapes. So this is again something which is interesting in terms of how this shape changes happen. Yeah. Yeah.
So the volume of the mitochondria.
It's okay. Yeah. Yeah, so the question is whether the volume of the mitochondria remains the same and the shape fluctuations are happening. As far as I can understand, yes, the volume remains the same. In fact, even when we are talking about the fission-fusion events happening all throughout the cell, there the volume, the total volume of the mitochondrion supposedly remains the same. So, yeah. Okay.
After the cell divides, you have interface, right? Essentially, this is when the cells are growing and so on. Then you likely have a mechanism whereby you produce more mitochondria, because otherwise you'd eventually end up diluting mitochondria every cycle, of course, yeah. So it's not a synchronized process like you have nuclear synthesis to be. So this happens across the interface is what's been hypothesized again. I mean, I don't know if there are any real studies out there. I haven't been looking very carefully. No, that is akin to fission, but you have biogenesis, as in production of mitochondria. Right.
Yeah.
Right. Yeah, no. You can have another fusion event with the one that just underwent fission for it to grow longer, but not like in the way that you, yeah.
The total mass of mitochondria must also increase, right? So mitochondria presumably should also grow in size and divide, right? So they also grow in size and divide, I presume, like bacteria.
Well,
Depends on the time scales. So during the time when cells don't divide, the volume of the mitochondria is the same. Once it divides, then they need to produce more of everything, including mitochondria.
Yes, at the end of the cycle, when the full eukaryotic cell has become two eukaryotic cells, at that time, between that time period, the number of mitochondria also need to double.
The volume of mitochondria.
And the mass, the biomass of mitochondria needs to double.
But
So at some stage, right? So therefore, both the numbers as well as the biomass of mitochondria need to double. So I don't see how it could be done unless mitochondria are also enlarging and so the cell growth basically. So how does mitochondrial growth happen? What is mitochondrial growth? Huh? No, wait. See, there is mitochondrial DNA which is there inside the mitochondria that replicates within the mitochondria, right? That replicates and right. So effectively, isn't it like a bacterium, which is living in some medium? Here, the medium is the rest of the eukaryotic cell.
Right.
But is it not a good model, the bacterial model for the mitochondrion? That the mitochondrion is a bacterium which has its own DNA,
right,
which replicates, and the mitochondrion expands, absorbing material, food from the environment, making itself larger, and then dividing into two mitochondria. Is that not the natural way of thinking about?
The division of the mitochondria is not separate.
No, that may be, division of, there might be any kind of mechanism causing mitochondrion division, and we heard a whole talk, right?
The addition of material happens at a particular time. Then after that,
is that okay?
No, I don't think so.
Yeah.
Exactly. Everybody should be able to listen to it.
Yeah, I think it's if. Okay.
Yeah.
Yeah.
Here's the mic.
Yeah. So, Apratim, you have to say something. Yeah, just go ahead. Yeah. Sure.
I just said, as Vaishnavi said, the mitochondrial cycle is independent of the eukaryotic cell cycle. Its synthesis of the DNA is happening throughout the cell cycle, and but a lot of the mitochondrial, what goes on to making a mitochondria is coded in the nucleus. So it's not like the mitochondrial DNA can code for everything that's there in the mitochondria. Most of it is in the nucleus, it's only a very small fraction of the stuff in the mitochondria that is coded by the mitochondrial DNA. So it has to be material, even genetic material, which is in the nucleus, which codes for mitochondrial proteins. So in that sense, perhaps it's distinct from a free-living bacterium. So DNA circular like the bacteria, right? Yeah.
Just a quick point. There is a mechanism known as retrograde regulation.
Sorry?
Retrograde regulation. When the mitochondrial content goes down, it turns on the transcription of genes required for mitochondria from the nuclear genome.
Okay.
So there's sort of feedback regulation there. So it maintains the mitochondrial content.
Okay.
You have this binomial model for redistributing mitochondria, and how do you stop errors from building up? And I was saying there must be some optimal number of mitochondria in the daughter cells, and if one of them ends up with too few, they'll just make more, so the error does not build up. Is that kind of okay?
We haven't tested that.
So that's good. Open questions. Right. Of course, again, this question about association of mitochondria with microtubules. This is a picture which kind of, this is labeled microtubules, this is mitochondria, and you can see that these are the regions where they are predominantly in, the mitochondria. So microtubules determine the unique orientation distribution of mitochondria in different types of cells. And of course, you have heard Vaishnavi talking about a great detail about this association. There's also localization of mitochondria, which is kind of understandable, of mitochondria near sites of high ATP utilization. That is not surprising. So this is one of the things that came up in different questions were being asked regarding the structure of the mitochondria. So this is the TEM image of the mitochondrion. And you can see that this is how it looks like. So you have this, what is called the inner membrane, okay? And then you have the outer membrane, and you have this different, this is the intermembrane space. This white region is the intermembrane space, and this is how it looks like. So you have these cristae-like structures that you have here. And so that I guess answers some of the questions that were being raised as to what the structure of the mitochondrion is. Okay, so moving on. Again, this is a question about fission. Yeah, sorry. Yeah.
Just a few days ago, a few weeks ago, there was a paper which showed that they are highly dynamic.
Mm-hmm.
So this was by super resolution microscopy. Pardon? By formalism, right? So the STED microscopy work which showed that that there's constant fusion and fission of these inner cristae also.
Okay, that's interesting. Okay, right. Okay, so so this is of course the question about fission and fusion, and this is of course this is a movie which was from V. Sriram's lab in NCBS, so which shows mitochondria, and you can see that what Vaishnavi was talking about. There's this different kinds of shapes of mitochondria that you're looking here, and there are regions of fission and fusion which are labeled. So that's MitoTracker, yeah. Okay, so the way the fission and fusion proceeds is, it has been also again discussed. So in the case of fusion, the fusion happens first. There's an outer membrane fusion that happens via these via MFN1 and MFN2. But also there is an inner membrane fusion. So first there is an outer membrane fusion, and then there is an inner membrane fusion. And this happens by this autotrophy, right? This is autotrophy 1, which causes this inner membrane fusion. And in fact, you can, if you have cells which are devoid of this, then you can have the outer membrane fission, but it never undergoes the inner membrane fusion. In the case of fission, what you see is that, this is for mammalian cells. What Vaishnavi was talking about was DNM1, which is in the case of fission yeast. In mammalian cells, this happens via this protein called DRP1, and these are receptors on the cells on the outer membrane of the mitochondrion. And this DRP1 gets recruited in this, and then it comes and binds and then forms this constriction, and then this fission process happens. Yeah.
So I think there's only, I'm not sure.
Yeah.
But probably there's just only one circular chromosome that's present in the mitochondria.
Mm-hmm.
So when the two mitochondria fuse together,
right,
what happens to the DNA, the other DNA? Right. So the two mitochondria coming together,
mm-hmm,
and now you have two circles.
right,
Coming together. So one
right,
gets disintegrated, or what happens?
I don't have a clue. Yeah, please.
Why are there so many receptors?
Well, I guess each of them have their own function. I don't know the exact functions of these receptors, but these are all apparently required for different stages of this fission event. So for this fission to proceed, you need each of, so each has its own requirement. They are required in one set, one kind of, some kind of cells, one of them is more, the requirement of one is more rather than the other. I think FIS1 is not required in fission yeast, or, yeah, exactly. It doesn't matter. FIS1, the presence of FIS1 doesn't matter in fission yeast, but it is required for mammalian cells.
So,
Sorry, quickly. There's more than one copy of nucleoid, I mean, more than one nucleoid can be more than one nucleoid in a mitochondrion. And when fusion happens, they remain distinct entities within this large mitochondrion.
Yeah.
In the three-dimensional image that you showed with the inner membrane.
Okay. Yeah.
So does the number of inner membranes, is there any significance? Because if there is more and if there is fusion, you're going to have many that are not going to bind with each other. So is there any significance to the number of inner membranes? And
so I think again, these large number of this cristae gives you more surface area. That is one of the things that is important. But again, as Madan was pointing out in this paper, it's shown to be dynamic as well. So it's I guess it's forms and new such cristae are formed. So so what happens when they fuse? I have no clue. Yeah, I mean, in fact, I don't think we have.
I just have a comment here. So, in fact, that's a very, very good point. The amount of cristae also increase in number when fusion happens. And if you think about just surface area to volume ratio, a smaller mitochondrion versus a larger one, maybe it would, one would think that a smaller one would do better. But in this case, what also happens after fusion is that the cristae numbers increase a lot. And the cristae numbers is directly correlated with increased membrane potential as well as ATP production. So, so there's a very interesting correlation with fusion and mitochondrial activity. One other very, very interesting thing is, for example, OPA, which is the fusion protein which he showed. OPA is present at the junction of the cristae. And at the lower part of the cristae, which is, inside of the mitochondrion, is the placement of complex 5 molecules. So the electron transport chain has many different complexes, and the complex 5 molecules are the ones which actually generate ATP. And so their presence, both OPA as well as ATP5, actually stabilizes the cristae. So there's a very, very, very interesting correlation between fusion, cristae stabilization, activity, and ATP generation. So membrane potential and ATP generation.
Can I just make a comment? That keep this between the two bilayers of constant. So imagine fusing. So imagine that you're looking at the fusion of two cylinders, and each cylinder is
built up of two bilayers, yeah, and you upon fusion you want to conserve the volume and the surface area, okay. Then you'll see that it's not easy to conserve both the surface area of the outer membrane and the inner membrane. Okay, it's a simple mathematics algebra, and then which means that you need to store some of the excess area in folds. So so just. I mean, this is without all the complications that you're talking about, which is of course added onto this, but just a simple statement like this would tell you that you need to have cristae and you need to have. Then of course biology does its own magical thing.
Yes.
I have a small confusion. So if you go to the next slide, one more.
Next.
Yeah, one more slide. Yeah, so this one. So the bottom one is always confusing me. I thought your mitochondrial membrane, so this mitochondria is like a tube. And if you cut the tube and look into it, that should be the cross section of how it should look from inside. Am I right or am I wrong?
Which should look from inside?
Yeah, so if you take a mitochondrial tube,
yeah,
what she was showing in her images,
yeah,
and if you cut it,
yeah,
and look inside, you will see the image on the bottom left.
Actually, you'll see this one.
Correct. So now if you go back there, so now when you talk about fission, I thought that the fission happens along the length of the tube, not this way, right? So this is the way you are doing it along the diameter of the tube. Or am I completely mistaken? So if this is the tube, right?
This is the tube. You have taken a cross section of that, and then yeah, and so
this is not proper representation.
The cutting is usually along the shortest axis.
This is cutting along the long axis, right?
So
no, I mean.
No, so this picture at bottom here.
It's a short one, no? It's the, if you look at it in an ellipse, right? Yeah.
So.
You're going to cut it like this.
Yeah.
No, I mean, no, it's fine.
What is shown in the longitudinal section
it's fine.
to show the inside.
Yeah.
It's actually cut. It is a transverse section.
I understand. So that the third picture in the bottom one is not
That's the correct one.
No,
That's right, no?
That's the correct one.
What's wrong with this?
We can discuss it.
No, it's not. It's
That's
not.
what it's
Yeah.
doing. So, when you cut this tube like this, so the
Maybe we'll go ahead and then we can decide later. So let's go into the question about microtubule based mitochondrial motility. Here, of course, we are this particular portion is regarding mammalian cells, where, as was pointed out by Vaishnavi again, that this is being done by kinesin and dynein motors, which are pulling on this cargo in two different directions. And this is also another molecule, which is another protein, which is basically can immobilize mitochondria in the presence of high calcium activity. So these have been discussed by Vaishnavi, so I'll not spend too much time on this. Rather, let's try to see how, so what are the ways, what are the things that we can look at in terms of how do we understand such systems. And one possibility regarding, for instance, if you look at mitochondrial motility, what we can do is, for instance, if you look at neuronal cells, and this is, these are neuronal cells, and what you are looking at is, again, in wild type, you are looking at the motion of mitochondria along in neurons. This is work by Steve Gross's lab in 2014. And then you can treat it with latrinquilin and to depolymerize actin filaments. What you observe is that if you do so, then the mitochondria move much faster because there is less crowding effect, and there's a situation where you can see a large number of mitochondria moving very fast. Now, what the kind of data that you observe, or the kind of data that they present is, let's say, how far does the mitochondria move as a function of time? What is the velocity of mitochondria, the run lengths? You can determine what are the pause times. So, for what instance, for what time scales does the mitochondria, while it's moving, it stalls? So all of these things are quantitative measurements that you can get, and one of the possibilities of trying to do a physical modeling of such systems is to for instance do something of this kind, which is like if you have a microtubule and you're talking about mitochondria as a cargo which is being driven bidirectionally by these two sets of motors. One set is a kinesin motor, the other is a dynein motor, which each of them is a similar scenario which I had pointed out in this where you have kinesin dragging mitochondria to the plus end and the dynein dragging it to the negative end. So you can try to write down master equation. You can approach it from master equations, where you write down equations for having certain number of so these are n plus and n minus would be having a certain number of kinesins attached to this cargo, and n minus would be the number of dyneins attached to this cargo, and you want to see what changes happens, the changes in the numbers of this kinesin and dynein that happens as a function of time. Where each of these quantities would be the attachment and detachment rate. So epsilon, so these are the probabilities, and this is basically telling you the rates of attachments or the detachments of the corresponding motor proteins that we are talking about. Okay, so now you can solve this kind of master equations and try to extract answer questions in terms of motilities of these cargo. You can also do, you can also, for instance, do molecular modeling of such kind of cargo being carried by molecular motors. In fact, you can extract all of these quantities, like the kind of stuff that we were just highlighting in the previous slide, in terms of what you observe in experiments, like pause times, run times, processivities, and all that. All of these things can be extracted, from a mathematical concept, from a mathematical perspective. Yeah.
I have a question.
Yeah.
So, in fact, when, especially in context of mammalian cells, what has been proposed is that for mitochondria to undergo fission, you need a pre-constriction step, which is mediated by the endoplasmic reticulum, the ER.
Right.
Because the DRP1 diameter is much smaller than the diameter of the mitochondria, where the rings are forming.
Okay.
So the pre-constriction step is mediated by the ER, and then you'll have recruitment of DRP1 to these sites, and then you have fission of mitochondria.
Right.
Could we envision a scenario in which, in this case, you have two motors of opposite orientation bound to the same cargo, which could be a mitochondrion?
Correct.
You could end up pulling mitochondria in opposite directions, perhaps leading to a pre-constricted state.
Yes.
Could you then predict what the fission rates would be in the presence and absence of bidirectional movement of mitochondria?
You can. I think it's possible to try to, no. So her question is whether we can, whether when you have, it's like a tug of war mechanism, not in this, writing down master equation. Yeah, not here, but we can in principle.
This model is a kinematic.
Yeah, this one is a kinematic model. But I think in general, I think, yeah.
You have to model the membrane and model this and stuff.
Not in with this master equation, yeah.
No, this is only about the motors. The mitochondria doesn't exist
Right,
yet.
right.
In this
No,
case,
I
there's no
understand
mind.
that.
Yeah.
So
I think she was asking about whether it can take it forward
in the future.
That's a tough calculation. That's not an easy calculation because you don't know the response of motor-driven forces on membranes. Meaning, there are n number. Let's say there's n plus and n minus. You don't know how load is shared. You don't know what is a
Mitochondrial shape transformation, when you have bidirectional movement, could you infer?
You can, of course, but you'll need to have. You might need some imaging of the shapes
Exactly.
of the as a
That's what I'm
function of
saying,
time.
Exactly. Yeah.
I'm just curious on a slightly different note. What is the space for modeling for mitochondrial biogenesis in relation to metabolic state of a cell? Because we're discussing lots about nuts and bolts, is tethering to microtubules, fusions, fissions, but there ought to be a feedback after sensing how much of it is needed, and then how much of membrane you make, internal, external.
Yeah.
Are those parameters on biochemistry available quantitatively that one can make use of, or no? So that's many of these things are very measurable nowadays to the very fine degrees.
I don't know the literature about this, but
I think there are very nice independent measurements of the biochemistry and activity and so on of mitochondria, but I don't know if there are many studies that actually link the dynamics and the
Do you know, by chance, the signal transduction pathway that feeds back onto the size of the...
Yeah.
It could be one of those receptors that you are decorating things with. You have shown one of those slides.
Sorry?
Several of those receptors, right?
No, I have no idea. Sorry. Anybody know?
Anybody else?
Okay.
So you mentioned you can get the information about processivity from, is this from this model or again from?
Processivity you can. Processivity you can from this.
But
yes.
processivity, is it the amount of time, the time spent by the motor on the microtubule?
Sorry?
The time spent by the motor on the microtubule. Is
Yeah.
It? So, but isn't it affected by the crowding and the things that are blocking the motors on the microtubule? And
So tell me once again, can you get an idea about?
Yeah. So it's the time that is spent by the motor on the microtubule.
Right.
So isn't it affected by the things which are present on the microtubule?
Correct.
Like
So you can
barriers,
have
so those
jams
things,
and stuff like that.
That information can be incorporated in this model too, is it?
It can be, yes.
Okay. Okay. Okay.
Sorry.
In fact, you can.
Sorry, there's no space yet here. This is just saying one cargo, a bunch of dyneins, bunch of kinesins.
Okay.
So you don't even have the notion of the length of the microtubule yet.
Okay. Okay.
But in this it is there actually not in the master equation, but when you're looking at it, when you're doing let's say molecular simulations, which is what is done here. Here you're looking at motion on a microtubule, and you have attachment detachments of. So there you can actually you can in fact have stochastic load sharing and all that stuff. So these data are extracted from those simulations.
From molecular simulation.
Yes.
So
one dimension. So you're basically looking at cargo being driven in the presence of certain number of motors. Yeah, on both sides.
So these are coarse grained simulations or actual?
Coarse grained, yes.
This particular master equation has these functions epsilon plus,
so these
epsilon
are
minus, etc.,
correct.
for which you will have to make some assumptions.
Yes. So, yes.
And these.
So these are for the molecular motors, of which a large number. I mean, you have a lot of information. So you know, for instance, what happens for kinesin, what are the attachment and detachment rates of kinesin.
And how they depend upon the existing numbers already, right? Because the epsilon plus, for example, depends upon n plus and n minus.
Yes.
These attachment rates, their dependence on the actual numbers already attached.
Yeah,
these are known, right? Or they can be phenomenologically determined.
Some of these, yeah. Some of them you can. I mean, so some approximations have to be made, but there are. I mean, these have been these kind of models have been done. People have been doing it for a long time. It's not. Where they have looked at tug of war. These are called tug of war kind of models, bidirectional transport, and how to go about doing it in terms of how do you get the parameters out of the use parameters from experiments, use them here and find out how it.
So basically, in this modeling, these functions epsilons and pi's.
Yeah.
They come from phenomenology, from experiment.
From experiments, yeah.
Effectively, right?
Yeah.
And you plug them in, and then you will be able to get
solve for this
this probability distribution of n plus and n minus. Now,
Sanjay, can I just
sure.
I mean, you're absolutely right, because for instance, you could say that the binding and binding is purely single molecular events, for which the parameters have been established experimentally and from in vitro studies. But as you are pointing out, that there's a degree of cooperativity here because of the existence of already motors. Now it's really difficult to molecularly model cooperativity, as you know. And so people have constructed logical models. And there are some with partial success and all that. There are people who have different models and so on. They come with their own set of parameters, which you try to fix by further experiments. So so I understand that it's not the best of right.
Yeah.
I mean, ideally, you'd like to have a theory for cooperativity and so on, right? And this is what you're driving at, I presume.
Yes, I mean, I just want to know what the boundaries of this kind of modeling is. What are the inputs and what are the outputs of this kind of modeling? That's all I wanted to.
Further, these models are kinetic in origin, so they don't have anything of the mechanics of the cargo, and it should certainly depend on not only the composition of the cargo, but the bending elasticity, the tension, etc., because this would then dictate how load is going to be shared by different motors. Yeah.
Right. So, and the actual movement, I mean, the last two pictures in the bottom, sample trajectories, that requires further elaboration of the model before you can get to the actual trajectories.
Right.
This is just the numbers of both kinds which are sitting on a particular cargo.
Correct.
But then, as a consequence of that, how will the cargo move?
Yeah.
That requires another set of assumptions and equations, right? So, fine. So you can get, so what you're trying to say is that if there are observations of how mitochondria are moving on microtubules, then there are mathematical models which can either reproduce or not reproduce that phenomenology, right? So are you saying that, has this been compared, let's say for mitochondrial motion on microtubules, has any such model been compared with experiment?
There have been, yeah. There have been theoretical studies with this, of this kind, which has been done, which has been attempted to find out how you can look at these kind of behaviors. So the experimental behaviors that you're looking here, how they match with
the actual experiment.
Yeah.
So this is the reference. This is one reference.
These are experiments. These are experiments.
No, does this do these experiments confront the theory with the experiment or not?
There's no theory. I mean, in this case, at least I don't know. I mean, they just talked of the experiment. I don't know whether theoretical attempts are made to explain these or observations. So that I don't know.
So since this is a meeting where experimentalists and theorists are sitting together, a rare occurrence,
right?
In this subject, I think it's worthwhile trying to identify which particular
regions or areas where we can,
both at the level of phenomena and the level of theory.
Yeah.
Where are the places where we can confront the two, where we can sort of have both and confront each other?
Correct.
So is this one place where, let's say, mitochondrial motion along microtubules?
Yeah.
Is this a place where theory can be brought to the point where it should be compared with the experiments, or
there's been lots of.
There has already been.
Yeah. So there's Steve Gross's group.
The existing phenomenological theories.
Yeah.
What is the status then? Do they agree with the experiment?
So, as I said, because it depends on your own microscopic model. So you have a microscopic model which will have some n number of parameters. I have another microscopic model which has some other number of parameters. Both of us will compare our models to the theory, to the experiment, and we'll get good fits by looking at it. So there are camps, but there are a finite number of camps. But there is a lot of work by physicists, biologists working together in this field. This is actually one of those happy occasions where the two communities have met. Very well.
Okay.
Right. What is revealing? So I think one of the things is what sets how are the different motors regulated. So for instance, Steve Gross is one of the persons who believes that that is the regulation of the kinesin versus the dynein that tells you the direction in which it moves and so on and so forth. Whereas some other group would say, no, it's actually forces. It's a tug of war between the two. For instance, Roop would think like that. And which one wins is set by the imbalance of forces. Yeah.
So, for instance, there are, in this one was, we were actually working on something which was to address something, a question about what is called the paradox of codependence, where it has been observed that when you mutate one, this is the traditional tug of war kind of scenario will ensure that, if you mutate one side, then the cargo will be dragged to the other side. But often it doesn't happen so. So when you mutate that, you observe that it doesn't really do that. So this has been, this particular behavior has been called as codependent behavior. So then for instance, in this case, what we were discussing was basically what we wanted to show was that if you assume that dynein, which Roop and others have predicted to have a catch bond like behavior, which is not a regular slip bond, which is like you apply a force and it comes off. It instead has a non-monotonic feature, so that you apply a force, it actually it binds stronger and then it peels off. So if you apply a catch bond like scenario to dynein, you can explain, hypothesize some of the behaviors that are observed in codependent behavior, in codependent transport.
So this is going too much into motors. Yeah, maybe if we come back to mitochondria, that'll be better. And I have a comment. Do we really need this detailed models for understanding mitochondrial fission-fusion? And we just speculate at a much higher...
Fusion. This is for transport only. So this is basically transport. So.
Yeah,
I also had that same question, that do we know the mitochondrial size distribution? Can we use fission?
Yeah, exactly.
Kinematics to understand
Right,
that?
right, sure.
And
right.
what are these size distribution? I would imagine that for fragmented.
Yeah, so I'll
have an exponential distribution for the fragmented ones.
Yeah, exactly. So
have a Paul law in the non-fragmented ones.
let's we'll just go into that. Yeah.
So then it will like.
Yeah. Yeah. Correct. So let me we'll come to that in a moment. So I'll just showing one of the possibilities of going into looking at these systems. The other, of course, is the very interesting behavior that you observe is called this mitochondrial segregation, where, as Vaishnavi pointed out, I mean, so this is again there's different phases of fragmentation, that is segregation, that happens. The mitochondria actually fuse to form these large. Initially, in the first stage, it forms these elongated structures, presumably because it requires a large amount of ATP for the process to initiate. And then it gets fragmented, and subsequently this fragmentation is divided into, this fragmented state is now divided into these two daughter cells, and these two daughter cells. And the question, of course, is how does this happen? Yeah. Yeah, I mean, so the point is that whether this distribution, I mean, roughly the number distribution of this mitochondria seems to be the same. So, so this is, of course, again, what Vaishnavi had also shown about how the cytoskeletal network is involved in this during this segregation process, wherein the actin cytoskeleton now plays a major role in doing this segregation. So mitochondria are in fact are on these actin tracks as they move from the mother to the daughter. So this is again one area where I don't know of anything mathematically which has been approached or to look at this system. So that is one possibility in terms of trying to understand how this behavior happens and how again in this link up with the actin with the cytoskeleton and the organelle can be implemented. The final stage, of course, I mean, the last thing that I wanted to discuss about was coming back to this fission and fusion, and this is some work which we had done about a decade back. I mean, it's not published, but we had started working on this problem, but then finally it didn't happen. It was with V. Sriram, I mean, who was then at NCBS. So this is on mitochondria. He was looking at Drosophila primary hemocyte culture system, and one of the things was again looking at fission and fusion. So this is experiments where this is a wild type cell, which of course shows so what is basically is these are images so that you can identify each mitochondria. So these are done so that you can identify each mitochondria. So that's the reason why you are seeing a fading off of the mitochondria as the time progresses. So this is wild type, and then so this is how it happens. So that you can to count. So we wanted to count the number of mitochondria. There was this question about finding out the some way of doing so. I don't know. I mean, somewhere if there's an yeah. So I guess they had perfected the system somehow, and this is with oligomycin. Oligomycin actually ensures that there is a large fission. So what happens is the mitochondria starts breaking up into smaller bits, fragments, and this is. So you can see that So again, these are images to ensure that you can observe. So you take it out of view. Basically, you take them out of view constantly as you are in time. Confocal stacks, yes.
It is also time.
I don't know. Probably these are Z stacks, because otherwise, in time, I don't know how.
In the beginning, you see all of it.
Yeah, well, somehow going out of focus. I think that's what they're trying to do, so that you can identify each of these mitochondria. Yeah. But the point is that in these cases, what you can do is basically, again, this is when you have reducing fission rate. So what you can do is you can reduce the fission rate, in which case you will see this elongated, really, large elongated structures. And again, this is a wild type, and this one is, of course, this large elongated structure, and you can see one by one, so this helps you in actually identifying each of this mitochondria, because otherwise the initial thing is like a big mess, and you really can't do anything about it. I mean, you cannot count, or neither can you find out the size distribution of this mitochondria, so the amount of. So what you can get out of these is to, so then we discussed, what we wanted to look at was an idea about looking at mitochondria as a system of undergoing fission and fusion. So so if you have so we started modeling it as a coarse grained system, where you have mitochondria which if they come and in a certain orientation, they will fuse. If they don't, then they will, they will fuse. If they don't come in that orientation, they will not. And then they will also, they can, it's a lattice model that we were looking at. So basically, it's on a two-dimensional hexagonal lattice with periodic boundary conditions. Mitochondria are distributed randomly at the sites, and each mitochondria is now characterized by its area. So imagine that the mitochondria that you're talking about, each of these mitochondria has a certain area, okay? And there's a certain orientation, so the mitochondria on the lattice will orient in a specific direction. And there's an activity parameter, which at this moment, let's forget about that, but there could be important implications of this activity parameter later on. So that was the hope, that we will be able to find out what that actually looks like. Now, in this case, of course, we took a hexagonal lattice, so it has its own 6 possible directions and all that. But the main dynamical rules of this is that there's a translation. So what you see in the cell is that if you're talking about wild type mammalian cells, then you're talking about mitochondria moving. You're talking about moving mitochondria, mobile mitochondria, and now you can think about mitochondria translating from one side to the next. It can rotate, so where there is a change, often you can see orientational changes of the mitochondria. That happens with a certain rate, and then you can have fission and fusion. Okay, so you have fusion, where when two mitochondria come, and then there's so a mitochondria hops and finds another mitochondria there, it fuses with it, and then you can have one mitochondria, fused mitochondria, and then there could be a rate of fission, which means that with a certain rate, it could just fission off. Okay. Now, how it fissions off? This is an important question. So the question is, if I consider it of a certain size, then where the chipping off will happen is important, because I can think of mitochondria as made up of several mitochondria. So that's like I can chip it off bit by bit. So what part of so does it fragment as break it up as in the middle, or does it fragment so that one part of this 1/3 of the length of the mitochondria chips off? That is something which we can control. In this system, this is, if people here in the audience, if they are familiar with the zero range process, they would identify it with this. So in this, what you basically know is that, and in fact, as was being pointed out, that you have, if I look at the probability of having a certain area, then this goes exponentially if you have a large fission rate. So if the fission rate, the rate of fission is very, very high, then you get an exponential distribution of this area. Whereas if you have a very low fission rate, then there is a possibility of aggregates forming. Okay, so this is of course a prediction of this kind of a theoretical setup, which was of course done. This kind of an system was given by Mustansir and Satya Majumdar back in 1994. And so if you now apply this kind of a understanding to mitochondrial system, then the question of course is that, so this is kind of the phase diagram that you're going to get, where you have a fission rate. Yeah.
So Vaishnavi, can one measure the distribution of the mitochondrial lengths and see if they're indeed exponential or power law?
Yeah, so I'll show it here.
Yes.
This is from our data?
No.
Okay. Not my data. We have a data.
Yeah. So this is what you will see. This is in the very large fission state. So you are basically trying to get, this is exponential. This is
This is an exponential.
This is an exponential because
Why are
it's
you plotting log?
yeah.
Okay.
Very few data, so you can't help. I mean, that's what we managed by that time. The amount of data that we could get.
So just one question. So when you have this experimental data, you have error bars associated with it. So
Yeah.
like if you
None of
use
this
a
is So these are very very preliminary data. After that,
No, I'm not talking about this data per se, but let's say if I want to get certain exponents or constants in my
Yeah.
model
Yeah.
from the experimental data. So one has to do this error propagation.
Yeah, of course.
When
you have to
this,
find out the error in the measurement of.
So how do this? I mean, are these error bars? I mean, when we incorporate these error bars in our model, do they also comply with the popular theoretical models like exponential decay or power law decay?
Well, that is kind of if you have a large fission state, then we would expect an exponential kind of a distribution of the sizes. So that is not something which is. I mean, it's kind of expected. Yeah, there's no.
So.
Yeah.
When you have error bars.
No, he's telling about whether you're measuring each of these points, whether it's something which is.
So there's a distribution.
Yeah.
My question is that you're getting this exponential behavior. Is it again associated with small fragmented microtubules? And if you get long microtubules, do you get a power law? Expected.
Remember what happens with the other guys.
It would be interesting.
Yeah. We have the data.
Does it show power
We have the
anywhere?
data.
Yeah, so I don't know whether, this is what we had obtained. So when this aggregate, that's how we could identify these aggregates. So this is P of the area as, yes, it's A, this is the same thing that we were talking about here.
I'm getting confused.
Yeah. No, it's so we could not determine that. That is something. That's what I said. That this is very, very all the data that we could get from the things that we got from the experimental side. This is what we could. This is the best that we could achieve. So, but at least there's some data which is like 10 years back. So
this is probably an example of never trust a theorist. We can fit anything we want.
No, it's not a fitting. Because it's all that. It's just getting the points.
It's plotted.
Yeah,
but it's plotted in the log scale to show an exponential.
I don't know why.
Why would you do that?
Yeah.
Exponential plot in the log.
Yeah, that's true. Maybe I have better. Maybe I should look back whether I have better data for this. But it's been a long time, so there's no way I can defend anything more than that. So, but at least I think what we had tried to establish a certain model for understanding the fission-fusion characteristics. This one? Yeah, so this is basically taking, so the question is, you can have a fission with a certain rate, then you have, so then this are, okay, so let me just remember what are the things that were there. Yeah. This is 99. Yeah, this is exactly the kind of the analytical forms of this is the diffusion, fragmentation, the chipping model was something which was this is what is called the zero range process. S is, let me check. Sorry, I have to. We can talk about this later. I don't remember exactly what are the different parameters. So I think this one is the chipping rate. Some source term, yeah. This is T. Okay, let me just. So we can look at it actually.
Which paper are we talking about?
I'll just bring up the paper and then you can see that. No, I think we can have more discussions about it, but that's all that I wanted to say in terms of looking at this.
So,
So maybe while he's looking at it, maybe we can think of open questions in the area that we can spend the next half an hour on. That'd be very great after this part.
Yeah,
so
if people have to comment on something, yeah.
So one.
Can the problem be posed like in the language of phase segregation? So looks like that one is sort of mixed phase where you have exponential distribution, another is segregated where we have large clusters.
Yeah.
Could be posed like that, some active form of that.
So
fluid phase segregation.
Fluid phase segregation.
Fluid. Looks like that all these filamentous structures, if I assume that they are like fluidic filaments and you diffuse around, merge, break.
Yeah, we can, of course. I mean, that is.
It's a possibility.
It's possibility, yeah.
So I have one comment about this model again. So you mentioned at the start that you use many of these parameters which go into this model from experimental data. Now, when it comes to experimental data, the data will have uncertainties in it.
Yes.
So that's why these parameters themselves will have uncertainties too.
Yeah.
So what I was asking is how this uncertainties in the parameter, I mean, how.
So for instance, for one of the things that what Vaishnavi was showing was fission frequency.
Yeah.
So that fission frequency is like a rate that will come up in the equation.
Okay.
So that is something which we can extract from the dynamics and put it back into the equations.
Okay.
So that is one thing that you can do.
So another thing that you mentioned was these models they show compliance with the experimental results.
Right.
So are you talking about qualitative or quantitative compliance? I mean, can we compare, say, certain quantities like velocities or certain forces?
Yes.
To the those computed using this model.
One of the things that, at least from the theory that I remember vaguely now, it's been a long time, but what we sort of we looked at, we said that if the fission, so one of the predictions that were coming out of it was that if the fission were to happen anywhere, so you're talking about a fission of the mitochondria, and the fission of the mitochondria, if it doesn't happen it's not symmetric, but it can happen randomly at any site. Okay. So at any place, let's say if you assume that the mitochondria is like a tube and I can have the fission at any point, then you would not, there's a possibility that you're not going to, theoretically, you don't see this condensation, this kind of an aggregate formation. Okay. So, and then you see just a power law decay without an aggregate. Okay. So that was one of the questions that we had at that point tried to look at as to whether we can come up with a predictive assumption as to where the fission is going to happen. So that was one of the things that we thought that could come out from the theory, which can be testified in experiments, that whether it's actually symmetric with respect to the mitochondrial structure, or it's asymmetric. Okay. Whether the fission happens in any, I mean, it doesn't matter whether it's happening everywhere. So
there's also a possibility that it could be a non-Poissonian process.
Could be, yes.
So is there any experimental?
No, I mean,
something that can.
Yeah, no. So you can see that. I mean, we had attempted it for about 10 years back now. So what the data I'm showing you is from V. Sriram's lab in 2007. So it's actually been more than 2007-8 data. So
Abhishek, what is measured experimentally exactly that constitutes the data?
So they were measuring they were measuring the intensity of so if you look at a mitochondria, well, there's a possibility that some of the mitochondria could also be out of focus. So a part of the mitochondria could be out of plane, and therefore we will not be able to look at it. But if I just forget about that and just look at the amount of when I fluorescently look at this, fluorescently label these mitochondria, then I'm looking the amount of intensity in this region of the mitochondria and finding out that as a measurement of the area of the mitochondria. So what I can do is do a so I can mark out the amount, the mitochondrial region by looking. That's the reason why those images where you could see them in, as they phase out, so was required for the identification. So you could identify one mitochondria and then keep looking at it and then measure the amount of area which is there in the mitochondria and find out what that, measure that area and look at it. Yeah. That was the experimental output. I guess that's the way you would do it, right? I mean, how we do what is the information that you can get from that is,
as
yeah. So that's exactly. So one of the things is overlapping. The other thing is, of course, as I said, is it could be out of plane, and therefore, if you look at the shape of the mitochondria, the first slide that I was showing you, the mitochondrial plasticity, which ensures that the shape. And we are looking at a 2D stuff, so we don't know whether there is. So you need confocal to really see whether, but that's like.
I was not confused because when you integrate the probability, you get the number.
Yeah.
Number is an exact measure of the number. So when you want to.
But even for number, you don't. There is a certain number. I mean, you even when Vaishnavi is talking about there's some certain. There's a large variation in the numbers. I mean, there will be certain fluctuations in the numbers that we are talking about as well. So identifying the numbers of mitochondria, again, will have the same problem that, it will be easier, of course, more, better than tracking the area, but it will still be a mess. As you can see, in some cases, it is very, very difficult to find out how many, because if they're in that aggregated state, it is difficult to find out what it is. So how many are in that fragmented state?
So,
hello.
I think that we are getting into a somewhat detail about in the discussion.
Yeah.
Perhaps we should come back to the general.
Sure. Yeah. Absolutely.
Phenomena, the biological phenomena.
And what can be.
And the kind of questions that can be asked.
Yeah.
So I don't know. I had one question. Maybe other people also have similar questions.
Then please,
Can you just plot down?
Yeah. So the first thing was about fission fusion and looking at it as a phase segregation model.
I would like to ask a question from previous presentation directing to Professor Anantha Narayanan. So he had mentioned that the partitioning error at mitosis. So I just wanted to know what it is exactly and how do you simulate that to get the data?
I can refer you to a very nice paper where they've calculated this. And what was the second question?
How do you simulate?
This was this was very simple. So in the mother, you have, let's say I started off with 1 mitochondrion, 2 mitochondria, 3 mitochondria. Independently, there's a 50% probability one mitochondria goes to the one daughter versus the other. Okay. And at the end, just see how many went to the mother, one daughter versus the other, and calculate the error rate. That's it.
Okay. The second question is in the context of curiosity, why mitochondria has chosen microtubules as a controlling mechanism for its fission rather than microfilaments or intermediate.
No, it's not filaments. True that it's only the microtubules. In this instance, it turned out to be rather simple. Okay. But in mammalian systems, there's a whole range of things that controls microtubules, mitochondrial fission. Okay. So, for example, I mentioned the endoplasmic reticulum a little while ago.
Yes.
The endoplasmic reticulum is known to affect mitochondrial fission. So does actin. So there is this, doesn't matter what the protein names is, but actin is also involved. Myosins are also supposedly involved. So there's a host of different factors that seemingly determine mitochondrial fragmentation. The question is, we don't know when what is used.
Okay, so the mitochondria is mostly associated with microtubules or it other fibrous elements as well?
Mitochondria associates predominantly with the microtubule, but it also has contact sites with the ER, which is where it predominantly supposedly undergoes fission.
Okay.
Other organelles also contact mitochondria, but predominantly it's a microtubule that it is moving along. It also associates with it, but there are also association sites between other organelles and mitochondria.
But the occurrence of the transport of mitochondria
is
through
on the
intermediate
filaments? No, there's no transport because you don't have, there's no known motor proteins of on the intermediate filaments. Yeah.
Thank you.
Okay, so actually, Abhishek, over here in this Greenfield model, the main thing is, from what I've been recalling, I mean, when the splitting is happening of the mitochondria, you basically have one mitochondria splitting up into two, which are of half the area, right? I mean, whatever volume that was there is half, whatever area that is there is half. Whereas over here, it doesn't seem that, in this particular modeling effort, if that is what is getting modeled over here, there doesn't seem to be any half factor popping up anywhere,
No. So here the point is that mitochondria can, in principle, in this case, we are not saying that it has to be symmetric. So we are not saying that it actually breaks off into half. It can happen that, a certain fraction of the mitochondria breaks off.
Yeah, but generally, in case we want to simulate something, I'm just talking about from trying to do this fusion, fission, phase segregation model. In case we want to simulate whatever is known, that it generally splits up into half.
That's the mitochondria doesn't. That's not.
Yeah. That's not true. So in fact, what we see is, especially in our system, I mentioned that the microtubule plasins are what are dynamic, right? So those are the guys that undergo depolymerization in the cell as well. So typically these are at cell ends. So what we actually see is that the shorter mitochondria so of course, it's not segregating equally first, but the shortest mitochondria are the cell periphery, which is where they have lost their contact with the microtubule network. So in fact, we do see it's asymmetric, it's not symmetric.
Oh, so the mitochondria itself doesn't separate into two halves which are equal in size?
No.
Okay.
So therefore, that's one of the things that was a question, that how it segregates, whether it's symmetric.
Okay, so here this thing is segregation is equally probable anywhere.
Yeah.
Okay, and that's what you're modeling. Okay, that's fine. Sure.
Exactly.
So I have some questions about fusion of mitochondria. So I guess these things have to come pretty close for
proximity,
to latch on to each other and stuff. And given that they are in a viscous, sort of cytoplasmic background, if you normally just think of two small spheres in a viscous fluid, they get prevented from coming close because of the background viscous fluid. So I'm wondering whether they need to be actively pushed towards each other, and whether that requires them to be attached to the microtubules in some way.
Yeah.
So and whether that's something that to think about.
Yeah, maybe Vaishnavi can, but I think you're right that in this case it has to. At least in mammalian cells, it is quite clear that they are associated with the microtubules and they are being carried by molecular motors, and therefore that is one of the active mechanisms which brings them into close proximity. So for the fusion event to happen as well. Yes.
So that means if they detach off the microtubules, another reason they may not fuse is just because they can't come close enough, other than the steric issues.
So,
in the case of
fissionist, I think it's.
In case of fissionist, we in fact had the same fusion rate in the absence of microtubules as well. So the difference there is though the cell is very small. So it's even the small diffusion that happens within the cytoplasm might be sufficient for these guys to encounter each other. In mammalian cells, it's a whole different ballgame. Yeah.
Perfect.
So, but in a way, it is different from, say, two spheres coming together in a viscous fluid, right? Because the lipid head groups are charged, so they will repel each other when they come into close proximity. So when, when vesicle fusion has to happen, the snare proteins would spend energy to bring those repelling. Here also, this OPA1 and they are all spending energy to actively bring about this fusion. So, yeah.
So you're talking about fission yeast, you're talking.
About everything, about fusions. The process of fusion requires energy.
Of course.
Yes, so,
of course. I mean, but bringing them close together, that's what was the question that he was asking.
yeah, but the thing is, even for
if you
bringing
brought them together, then you'd still.
meaning they wouldn't like to come together because the head groups of the lipids would repel each other, right?
Then you require the MFN1 and all these things are required for all these processes. Yes.
So if I can get back, I mean, I have some very naive questions. Do all cells have mitochondria doing fission?
Yeah.
So it's not a universal phenomenon for all life. Why do they do that? I mean, the ones that do fusion.
Yeah, I think this is a very important question.
Otherwise, we keep going back and we can do.
So this is actually a general question. I wanted to get a perspective on why there is mitochondrial fission and fusion. Of course, you need fission because mitochondria will grow and you need to divide and have more mitochondria as a cell is growing. You presumably need fusion also because for some reasons, okay? So I think why is there mitochondrial fission and fusion? That's one generic question. And what are the quantitative features of fission and fusion that are required? I mean, are there certain characteristics quantitatively that fission and fusion ought to have in terms of their rates, in terms of the eventual size distribution or whatever? Is there some generic property that one can put one's finger on, that this is the desired property of the fission-fusion process or desired properties that we want out of, is there something that one can put one's finger on?
Yeah.
So these are two specific questions, but maybe, can I ask both the speakers today to write down two broad questions for the audience, right, in the subject, according to their perspective. What are the two...
Sanjay, one of the questions that you asked is a very relevant one, whether across mitochondria, I mean, this question about, I mean, if I can say so, universality. I mean, whether you can say, look at different sets of cells, a large number of them, and see whether you can say that, okay, this has some distribution of mitochondria, this has a certain distribution of mitochondria in terms of the sizes of mitochondria, and also look at whether this question about what we can understand about fission and fusion. I mean, why is it necessary to? How does it keep that distribution? I mean, why is it necessary to have that? That is, I think, I don't know. I mean, I don't think it's an unanswered question.
Yeah. So adding to the list of questions, the fact that mitochondria are concentrated around microtubules, is that also something which is reasonable and quote unquote required? What is the reason for this kind of localization? So
that I would, in a way, sorry.
So maybe I should now give the mic away because. Can I? Broadly, what I would like is a set of questions that one can, take away from the session as questions which are perhaps overarching questions in the field, to which answers are not that clear. So if something like that can come out from the discussion, that would be very helpful as a takeaway from the session.
In general, I guess organelle transport of any kind, I mean, this active, requirement of motor-driven transport is something which is seen in a large number of organelles, right? It's not only for mitochondria. It's seen for lysosomes and, that's generic. So that is something which is kind of, the fact that the organelles would want to associate with mitochondria, with microtubules, I feel is, that is required, right? Otherwise, to move from one region of the cell to another, which is requirement of energy. Just naively, if I think about it, a certain region of the cell requires some process to happen, and it require more energy for that process to happen, then a diffusive process to go and reach from one corner of the cell to another would be much and in fact it has been some yeah.
For small cells, diffusive transport is good enough. Even then you find them clustered around the mitochondria.
That is true, yes.
Can I make a point? Just an amendment to what Sanjay said, which is, I think the fission part is easy to rationalize, if not understand, because it's a concomitant of reproduction and the fact that you have two where you had one. It's the fusion part which doesn't immediately yield an explanation.
Increase.
So as much as fission makes sense to divide mitochondria and also to distribute genomes into two new mitochondria and maybe even discard the spoilt mitochondria, dysfunctional mitochondria. Fusion is very important for complementation of mutations as well. So that has been shown very, very nicely. If fusion does not happen, then mutations do not get complemented. So at a genetic level, complementation of mutations is one important reason for fusion. So then the entire mitochondria, even if it has few genomic copies which are not okay for some genes, will actually be healthy because of complementation. So this is one point. And so, yeah, sorry. So we are talking about genetic complementation here, which essentially means that if you had, let's say like in chromosomes, we have two pairs of chromosomes. And if one gene is mutant in one, if one gene is mutant, whereas the other supports the wild type function, then you really don't see the mutant phenotype. So fusion allows for complementation of mutations if there is DNA copy, mitochondrial DNA copy, which is aberrant. And if another mitochondrion actually has a healthy DNA copy, then you will see that the mutation effect phenotypically gets masked, gets suppressed. All right. So the functional unit, mitochondria is actually a functional unit. So fusion assumes a very important role there, and that has been shown in quite a nice study. So this is one role for fusion. The second part is about biogenesis. And so it turns out, so if we just take the context of stem cells have very weakly developed mitochondria. They have typically small round mitochondria. And these small round mitochondria do not have very elaborate cristae in them at all. So in fact, what is thought is that many, let's say mammalian stem cells, they are present in a niche area which is glucose rich. So it is getting the energy that it wants. And so, for example, the rho zero cell, which Vaishnavi was showing, it's very nice to correlate what we see in stem cells to something like the rho zero cell, where weakly active mitochondria can be supplemented by just glucose from the environment. Okay, when a cell differentiates, and which is akin to a lot of cell culture experiments, mitochondria are elaborate, of the kind the pink picture which she was showing. They are elaborate. They are present all over the cell. The cells are huge, and fusion plays a very important role to form that big structure. And fusion has to be, so fragmentation has to be increased in those cells during the cell cycle for equal segregation. So this is a very, very big generalization. I mean, from our own experiments, we see that different cell types in an animal tissue actually handle mitochondria very differently. So at the very basic level, this is what fusion and fusion is about and what fission is about. So then there are complicated experiments. I think let's not go there. I mean, the surface area of the outer membrane surface area when it is fused could actually have a certain membrane potential to dock on them a certain set of proteins and so on. and that may increase in activity, but it's too complicated. I mean, I don't know. There's not even very good evidence for it. That's
so for instance, even in the when they're showing this,
actually, I want to add to ma'am's note that the protein part is also, I think, very important, because if one, as you said, that the mutation will be masked is if two mitochondria get fused. It might so happen that the proteins, if I have some non-functional protein in one mitochondria, and if it fused with the other mitochondria, which has the exactly what function they should do, for example, the complex 1, 2, 3, and 4, and the 5 ATP synthase, these proteins, if some mitochondria has non-functional protein, when it gets fused with the other mitochondria, with the proteins, with the proper activity, the whole mitochondria, the fused mitochondria can act better, I think. So with the genetic element, the protein part is also very important, I think, for the fusion.
So in all of this, so the mitochondria, when we study, it's the powerhouse. We have not discussed anything about the energetics and so
and all that.
So does that play a role in all of this fusion and fission and so is there anything known about that but is there something more known? Like in this case, you said that fusion plays a very important role in complementation of mutations, etc. So is that?
Fused, like I said earlier, fused mitochondria are also high in membrane potential and are also thought to be more active. They are more resistant to autophagy for degradation, essentially. Yeah, they are. They have more cristae, and
I mean, this opening and closing of calcium channels. I mean, this
they are better storehouses for calcium.
Yeah.
Right. So in other words, can the cell use this as a regulation to, for the energy, to control the energetic requirements?
Like something which is,
I don't know. I mean, I'm just.
I guess
we haven't discussed.
Big experts on this. That's
no, but I'm just bringing it up because we have not discussed this at all.
We are looking at it
simply as some membrane compartment.
We are. I guess we are looking at a complete mitochondria rather than looking at each of the an individual mitochondrion and then looking at the internal details of it. Yeah. But that's because I guess none of these here are exposed.
So,
Yeah, that is true.
Do they also localize in regions where maybe energy will be required more? Maybe, let's say for a crawling cell, wherever actin polymerization happened, do I see like more
Right. So that
mitochondria localizing there?
Yeah, that, if I remember, I was showing you a plot of where in cardiac muscle cells, I think.
So, for instance, growth cone of axons, do I see?
Yeah. So this is like, of course, this is known. So localization of mitochondria near sites of high ATP. So this is, of course, a schematic, but you know that this actually happens.
But these were boost.
Sorry. So this is sperm tail. And this one is cardiac muscle. So this is where it. So actually it does. That part is actually well known. That localizes near regions of high ATP.
So we can bring back. So what we're trying to look at is a generic process of fusion and fission. Forget mitochondria. Are there other processes in biology where we see fission, fusion, and are physics the same there?
Right? I mean, organal.
For example, Shashi has a system where there are mixococcus things going along a line. Lines are microtubules. Do you see fission, fusion? What about distributions of sizes there and so on? Can one think about things? Endosomes, for example.
Endosomes, yeah. I think
so. The question I'm trying to pose is there something specific to the mitochondria microtubule system, or is this a generic process of fission, fusion that we are looking at? And if that's so, there are a lot of the physics of fragmentation, aggregation is known.
Already well known.
In a very, very well established manner.
In fact, yeah, that's exactly.
Books and books written about those things. So question is this something specific which is driven, active, has some functional thing which is not incorporated in the earlier physics pictures? Or is this a generic fission, fusion model that's...
pointing out, right? That if you're increasing the length of the microtubules, you're basically getting longer mitochondria. This thing is not going to explain that.
Yeah.
So that is something, like Madan was basically saying that might be the reason you're basically having larger lengths and implying larger mitochondria is because, you're basically having more molecular motors, which are basically causing the fission. But I'm not sure whether, I'm not sure I understood the argument, but the thing is, it still seems to be a very interesting problem as to how long lengths correspond to longer mitochondria. And was that something to do with generic thing in biology per se?
No, maybe a better way to answer it is to rigorously compare physics models with experimental data.
Yeah.
And then say that there is new physics. But otherwise it's just words and keep going.
So I can see one place where the usual physics models will be inadequate. Because in the process of mitochondrial growth, there must be, in addition to fission and fusion, another process that causes the increase of mitochondrial biomass.
Yeah, that's
why? Because both fission and fusion conserve mitochondrial biomass. So what is the process by which mitochondrial biomass increases? This is a growth process. This is a kind of a growth process which must exist. And we've only talked about fission and fusion here, but there must be another process which adds to biomass, mitochondrial biomass inside the cell. What is that process? What do we know about it quantitatively?
I think in addition to that, I don't have an answer to that, but in addition to that, you need to make sure that between one division cycle and the next, you actually bring up mitochondrial mass to what it was in the previous division. So there needs to be a control over how much mitochondria you make as well, right? Because technically the way it would proceed is that you essentially bring it up to the same amount that you had in the previous cell division. Otherwise, you'd end up diluting the mitochondria. Yeah.
So you need to, if your cells are going to, you're going to get 2 cells at the end of the day, you need to double the biomass of mitochondria.
Yeah, so to do something like that and understand biologically, I think you need systems where, other than fission, there is mitochondria, where there is 2 or 3 copies of mitochondria in the system, and they're also well correlated with the cell cycle and everything, because otherwise it becomes too complicated to even understand. Exactly. Yeah. So are there systems in biology where there is single mitochondria or 2 mitochondria which are dividing in concert with everything? Like chloroplast, yes, definitely. There is a single chloroplast, and coupled with the nuclear division, there is also a division of chloroplast and everything. But mitochondria, are there species that have single mitochondria are still living? That might be interesting systems to look at these things. Single mitochondria.
And there are
Trypanosoma also, right?
And there are
So here, when you looked at the phase, even the mean field model that you had proposed, which was Satya Mazumdar's model, over here, fission and fusion, they're almost happening simultaneously. Whereas, I had also talked to Madan, and he basically said that, there might be specific times during which there is more fission happening. There are specific times during which there is more fusion happening. And that thing could basically change the kind of whatever thing that you're basically modeling, giving you new kind of effects.
Sure. I think that model was, the model that we were looking at was a first attempt.
First attempt, yeah, I understand that, yeah.
How we can, because there is, there has been, at least in the context of mitochondria, we didn't see anything to approach even.
No, I understand, but when it was said that if it was just like a growth model or aggregation model, this thing doesn't seem like to be normal aggregation or normal thing on which just papers have been written. These are specialized, happening at different intervals, fission and fusion, and how does that basically correlate to actual biological phenomena, whether that thing is basically correlated in a way which is going to be required for cell division in some particular aspect. That thing is like a fundamental question in case
I have to say that even I was, since after, for the past few days, I was trying to look at, have there been any approaches because it's been 10 years since, when we were looking at this. And yeah, exactly. Yeah, absolutely. So that is, and the point is that there has been, I mean, Vaishnavi can correct me if I'm wrong, but to date I don't see anything in mitochondria, even for even using such a simple model of fission fusion and trying to understand what type of size distribution of the mitochondria can happen, and looking at it both from experiments and theory, that to date doesn't exist. I mean, there has been no such paper which I have seen where people have looked into it.
And are there timescales over here, right? Fission happens or not?
Even that seems like a first step, whatever direction we are talking about. At least as a first step, also it's something which could be looked at, and then of course, I mean, as people have pointed out, there are very important questions.
No, but experimentally too, like the time series of the fission and fusion events and the time distribution.
Yeah, and that is not true, and that is absolutely not difficult to get. In fact, you have seen Vaishnavi's data where she already shows fission frequency. I mean, there is already a data of fission frequency which will give us a fission rate.
Right.
So that rate, of course, comes into.
Yeah, but as a function of time, that's the main thing. Yeah.
Yeah. Yeah. Suppose it is not. Of course, then of course that's an additional incorporation into maybe what we are looking at, and in terms of model, and maybe that is going to modify.
Coming back to earlier question as what could be different from simple physical systems of aggregation. These are also active processes, so there are active mitochondria being actively transported, which affects both the fission and fusion, and as opposed to just passive diffusion. So that again may show up as differences in the biological.
The point is that what I don't understand is how does it affect fission fusion? I mean, we can of course think about it as, let's say, it's bringing them, maybe they will bring them closer together faster. That is true. I mean, right? But here there is no. Yeah.
So even if you don't have passive diffusion, you can write down simple models that break detail balance. Maybe that's enough. You don't need to go into the details of whether it's motor driven or not.
Right.
That may not be necessary at this level of modeling. So,
can I?
In the fission yeast, like the mitochondria are on the microtubules, right? So what instead of looking at the fission rates of the mitochondria, you look at what happens if you break the microtubules. Can that somehow give you some idea?
Yeah, that's the first experiment we did, right? So if you depolymerize the microtubules, then you have complete fragmentation of mitochondria. We measure the fission rates of that in the final last but few slides.
Okay.
That experiment was shown that when you have.
So I have one small comment to add to the comment that you made regarding growth. So indeed, also other organelles like ER and Golgi also have to double, right? But I guess the point here is that there is a mitochondrial DNA, and the question really is that being used to manufacture or do something more in addition to what the host is doing? I guess that's the point, right?
The question on biogenesis, there may be a possibility of extracting that information from papers, because there are clear mutants that actually prevent biogenesis. No, there are clear mutants, let's say, which affect the increase in the entire electron transport chain machinery, almost. And to compensate those mutants, there are two changes which take place. One is that the transcription of all of the proteins increases manyfold, and I'm sure it's possible to get estimates on how much it increases in the mutant. And there is also an increase in the amount of mitochondrial DNA. So so using this mutant, one can also compare what's happening in the wild type scenario. So it allows one to follow the question of biogenesis with respect to shape as well as the entire machinery. I think there may be literature which compares two scenarios where biogenesis takes place. and has also looked at increase in protein content and the rate of increase in protein content. And that may be a very nice starting point for this.
And these remarks. An experimental question. Is it possible to observe a mitochondrion through time and see a change of size of that mitochondrion? And and this is the change of size caused not by fission or fusion, but by whatever other processes. That is, if a fission process happens, that is separate, that can be identified. If a fusion happens, that also can be identified. But without fusion or fission, is there a change of size of mitochondrion, and can we observe that? The reason I'm asking this is that for bacteria, there are now single-cell experiments which actually study the time, the size of the bacterium as a function of time. And those studies, and as to when it divides and when it, etc., those are very revealing in terms of the dynamics of the bacterial physiology. So the physiology, so the mitochondrial physiology within the cell, whether we can study that experimentally. That's one question. And there are, both the energetics as well as the genetics of it, and the generation of proteins which is required for this kind of thing to happen. There are both experimental as well as mathematical models for bacteria now. And it might be interesting to think about whether such kinds of things can be done for mitochondrial dynamics. You said that you can study?
Not so. The biogenesis would require you to have the way I think about it in my head is for you to already have a marker for the existing mitochondria, and you would have to have a marker for the biogenesis. But what specifically will mark only that region of mitochondria that has been added newly, I have no clue. I have no idea. No, size is one thing, but size can also change. So you'll have to have not just one single mitochondrion labeled, but you also, in that instance, you'll only have to have one mitochondrion labeled. Because fusion can also lead to changes in size.
Can you not work with cells that have only one mitochondrion?
Exactly. It's complicated.
Can you not work with a cell that has only one mitochondrion?
That's plasmodium, and I don't think I have the facilities to do it. Yeah. You can switch it off, but then I don't know if that will reflect what would normally happen in a cell, because you're artificially forcing something to happen. You can measure in that instance for sure. That's definitely there.
So if you find
Still,
that
it's hard. It's very hard.
Yeah. No, the point is, I think
blobs of mitochondria that is ready to die. So
but I think you don't need.
But you have a movie, right?
So for instance, if you look at a complete set of all these different mitochondria, you can pick one of them which is not doing the fission fusion and follow it in time. And
so I was thinking that you have a movie, you can follow them and.
No, it can, but if it does it, then you don't. I mean, you reject that. You try to see if there is at least one which you can find out of a set of, I guess, tens of mitochondria.
Until they merge, until you see fission and fusion event.
But I think Sanjay is also
follow it up to that point, right? And get enough statistics.
To look at it at a
Doesn't
really
that
long
work?
time. Isn't that what you're trying to? Not necessarily. Then of course I think. Then of course 20 minutes. Yeah. Then I think then that is.
Order. I think we'll take the two questions from the students there, and then we can wrap up. We can come back after.
Go ahead.
Simply label a mitochondria and then study it for a longer time. Then if the size is increasing, so other biomolecules are getting added to the mitochondria, then the fluorescent intensity should decrease with time.
See, the other thing by looking at those movies, because I recalling from the days when I was looking at those movies, a lot of times I would see that the mitochondrial shape would just change, but that's not because it has actually nothing has happened. It has just moved off plane. I mean, so you're looking at it on that, and it has moved. I mean, now I don't know. I leave it to the experiment. How do you? I mean, in time when you're doing it, and then it just moves out of plane, then you have to. Actually, it happens a lot.
But can you answer that, Vaishnavi? Can you just answer it?
Yeah, no.
No, okay.
No, the problem with this, it's imaging over time has its own caveats. So if you image for more than a few minutes, you also have bleaching. So you'll have to correct for that. And you're not sure how much of the effects you're seeing is caused due to bleaching or if it is due to addition of new material, which is unlabeled, like you're suggesting. But it's a good idea in theory. Experimentally, some things are easier than others. This is not an easy experiment to do.
I have a question regarding one of the previous, I think a few answers before it was taken that, like for example, in the cardiac tissue, you see that there is more mitochondrial localization, right?
Yeah.
Right? So my question here is it known whether the number of fission processes versus the number of fusion processes is...
There at that, I mean...
like the ratio of number of fission versus the number of fusion processes, is something known about that?
I mean, you're saying where, I mean, in
the tissues where there is more energy requirement, for example, the muscle tissues or the cardiac tissue.
You're saying whether they're more fragmented or they're in the more...
Like more fusion events or more fission events?
Well, I mean, one of the things that I showed, if you remember, during this whole breaking up of the cell into two, I mean, the initial stage, the G1 phase that they talk, there presumably the mitochondria initially start off by becoming very elongated because the requirement of ATP is more, and apparently that helps when you have elongated structures, elongated mitochondria to have.
I mean, more surface area.
Surface area in that sense. Here, of course, this is of course something which is, I don't know whether it's this one is a schematic, so it's highly unlikely.
The last question, Srini. Let it be a broader thing, not
Yeah, I don't know if it's broader or not, but the thing is, what if there was a way to take off this mitochondrial attachment to microtubule and attach it to something else? Like the mitochondria microtubule link is completely disregarded, you make it free of that, and with some other filament if you attach it, will it give any additional information on the fission fusion? Because then you don't have the complication of the microtubule dynamics and the cell cycle and everything. Say I had it completely independent of the microtubule filaments. I can link it to some other filament in the cell. So will it help in any way or not?
Okay, I'd argue that in this instance is actually employing the microtubules to maintain a balance between fission and fusion. It's not that it's a process. So, okay, let me put it this way. Microtubules are undergoing their own thing. I mean, they're doing whatever they do, right? Depolymerization and polymerization. And that is essential, I think, for the mitochondrial morphology in these cells that are non-dividing. Because without the microtubule attachment, they don't have, I say that these are cues for initiation of fission and fusion. So attaching it to another substrate would possibly prevent the fission, but whether that would be beneficial for the cell is unknown to me. Right. Sure, but it then comes back to whether these guys, even though they might be less prone to undergo fission, are they fit?
Yeah, are they fit or not?
Yeah.
Up to 7 you have free time. You can go around the campus or maybe take a cycle and go out or do whatever you want. 7 we have dinner, 7 to 8:30, and 8:30 for people who are interested, we assemble back here and we'll have a discussion, particularly with the students and the postdocs. I hope that some of them will come back and take more charge. So the discussion will be about the two topics that we have talked about today. So it's more like a summary kind of a thing.